Welcome to Our Connection, the podcast that brings together patients, caregivers, researchers, and innovators involving to to solve the challenge of rare disease. Today's guest, Dr. Stephen Quay, a physician, scientist, biotech entrepreneur, and CEO of Atossa Therapeutics. Dr. Quay has spent decades at the intersection of biomedical innovation and oncology and drug development. His company is advancing endoxifen, the active metabolite of tamoxifen, as a next generation endocrine therapy designed to eliminate the metabolite variability that can eliminate the traditional tamoxifen treatment. Beyond oncology, this work is now expanding into the rare disease space. In January 2026, the FDA granted orphan drug designation to Duchenne Muscular Dystrophy. Based on research suggestion that endoxifen may help increase production of utrophin, a protein that could help compensate for the missing dystrophin seen in Duchenne. Today, we're going to talk about the science behind the approach, what orphan drug designation means, and how innovations developed for cancer sometimes end up helping rare disease communities. Dr. Quay, you have experience as a physician, scientist, and biotech CEO. What originally drew you to drug development?
Well, that's a long story, but I'll cut it briefly for your folks there. I grew up in Michigan, and kind of always wanted to be a scientist from probably 10, 11, 12 years old. Now, my dad was a high school chemistry and physics teacher, and that's probably where I got the inspiration of going in the lab on weekends and watching him prep for classes. Now, he had a heart condition called rheumatic heart disease, and his vision was that I would grow up and become a doctor and stay in the small town that I grew up in and sort of take care of him, I guess is the best way to put it psychologically. At the time, I didn't realize it. So, being a dutiful first son, I combined the two at the University of Michigan. And so I got a PhD in chemistry to satisfy my scientific curiosity and an MD from the university there. And then went on to do a postdoc in chemistry at MIT with a Nobel laureate, a residency at the Mass General across the river, Charles River from MIT. So I was a doctor during the day and a chemist at night at MIT. And then taught at Stanford for about a decade when I invented the gadolinium that's used in MRI imaging uh, and commercialized that and now have six FDA approved drugs, under my belt. So I always gravitate toward the science, but I do want to thank my father who's passed away now, but for sort of getting me into the mode of getting an MD because I think the medical degree doesn't help you do science, in my opinion, but it helps define the problems you should be looking at from a PhD scientific based format. So, now 7 FDA approved drugs later, I started Atossa Therapeutics with the model of preventing and treating breast cancer, but taking the drug that I invented, endoxifen, wherever it goes. And quite remarkably, in the last 2 years, its ability to treat two very different but mechanistically similar rare diseases has got me really excited to be in this space. And while I'm new in the rare disease space, I've been to some meetings with parents and with advocates and that sort of thing. And it's really a remarkable area of drug development and of medicine that I heretofore had not been involved with, but am really excited to be hopefully making contributions to.
Yeah, it's exciting to see all the new drugs coming out now because of it seems to be a big season for new drugs. But 95% of rare diseases don't have an FDA approved treatment, and so this is kind of exciting times. Um, many people living may not realize, many people listening may not uh, realize how many drugs come from oncology research. Um, why has cancer research historically driven so much innovation in medicine?
Yeah, I think it's hard to pin it down to one idea, but let me talk a little bit about the intersection of two things that I think are important, which is one is, oncology is based on the principle of mutations that typically happen spontaneously, so they're just mistakes. Every time a cell divides, it has to copy 6 billion letters correctly. The first pass, it makes about 2,000 mistakes, and then we have about 30 different enzymes that go in there and dutifully repair the couple thousand mistakes down to zero, hopefully. But every now and then, one persists, and it persists in a spot that can be detrimental. Most mutations are not detrimental; they're almost silent. But we have some oncogenes, genes that drive cancers and things. So the fact that oncology is based on genetic changes and many diseases have a genetic component, I think that's pillar number one of why drugs come out of oncology. Pillar number two is that we now know that many aspects of oncology are driven by both metabolism and inflammation. When I say metabolism, I have to go back to the 1920s, so we're now 100 years since a German physiologist named Warburg first identified that cancers, unlike other tissues, could only burn glucose. They couldn't burn other kinds of fuels. And so this metabolic aspect of cancer, of course, has a commonality in diabetes and obesity and other conditions. And so there's a thread in oncology that leads to those other chronic diseases. And then finally, inflammation itself. My theory on why it's involved with cancer is that basically it makes cells grow faster than they'd like to grow. All cancers start with normal cells and go through two phases of what are called hyperplasia, where there are too many cells. Normal hyperplasia and then atypical hyperplasia, where there's a few mutations that make the cells look funny. So if inflammation is driving cells to grow faster and those 30 enzymes are not repairing the 2,000 mutations that occur, that's probably the basis for why cancers arise in inflammation, or why cancers arise in metabolic disease, or why cancers arise with genetic conditions. So all three of those pillars make cancer drugs good targets for off-label use, for ancillary use in other conditions.
Hmm. Yeah, it makes sense with how fast they grow. It's um, let's see, let's start with the basics. What exactly is the endoxifen, and is it related to tamoxifen?
Yeah. So again, just a little bit of history. So we had World War I, where the chemists contributed mustard gas and other chemical warfares. We now have conventions and treaties to not use those. But nonetheless, a man named Dana-Farber in the late 40s said, I wonder if I could rapidly, massively reduce the dose of these toxins that were killers during World War One, and treat cancer. So the 50s was the introduction of chemotherapy, where unfortunately the challenge was doctors would try to kill the cancer but not kill the patient. And the better the doctor, the closer they would get you to that threshold of almost killing you. It was very harsh treatment, very harsh treatment. So in the 70s, a woman chemist at ICI in England was studying ways to potentially create a birth control pill, and she created tamoxifen. She was doing some animal testing with it, and found that it actually inhibited cancers, and specifically estrogen-driven cancers, specifically breast cancer in her models. She spent a lot of time in the lab proving that, and a lot of time in the boardroom getting the company to continue development. If she had not been so persistent, tamoxifen would have never been developed for cancer at all. But it was her tenacity that led to both the scientific discovery of what tamoxifen was doing and the drive to get a large, massive company like a big cruise liner to pivot into cancer therapy. But it was finally approved by the FDA in 1977, and as they say, the rest is history. It is the most used specific drug for cancer. And it was the first model of not just simply trying to kill cancer cells, but to work with the inherent metabolism of the normal body of the cancerous cells and manipulate those metabolic pathways in a way that was useful. So but what she didn't know, she couldn't have known because of the tools at the time were not available, was that tamoxifen actually isn't a drug that kills cancer cells. Excuse me, it is a prodrug. You take it, it goes through the bloodstream to the liver, and the liver does two kinds of chemistry on tamoxifen. It adds a hydroxyl group to make it more soluble, because that's what the liver does with every xenobiotic or foreign chemical that it gets. When you eat all your vegetables and you get all your phytochemicals, the liver's trying to get rid of them by making them more water soluble. So the first step is putting a hydroxyl group on, an OH, to make it more water soluble. The next is to take a methyl group off, which makes it more water soluble. Those two steps lead to a chemical called Z-endoxifen, which is the total efficacy of tamoxifen in breast cancer. With the discovery 10 or 15 years ago of that new principle, we jumped on that and said, let's develop just endoxifen. Because when you take tamoxifen, you're making 20 different chemicals. Endoxifen is one of them, but there are 19 other things that the liver does with tamoxifen, and a lot of them have mischief. So many of the side effects of tamoxifen are related not to the endoxifen itself, but to these other 19 chemicals. So giving endoxifen in pure form lets us go to higher doses and get better efficacy. And fortuitously, I didn't, I'll take credit for making it more effective, but I won't take credit for the side effects because you don't have the other 19 molecules. It's a cleaner drug and it has a better profile. So for about 6 years, we've been working diligently in preventing breast cancer and treating early breast cancer in the clinic, phase 1 and 2 clinical trials in that area. But then about 2 years ago, using generative AI, so this is a wonderful story of where this new thing we call AI can impact drug development. I brought that into the fore about 4 years ago. About 2 years ago, generative AI came out with an analysis which basically said, hey, Quay, you got to look at what endoxifen does in this very strange protein development pathway called the utrophin dystrophin pathway that led us into the work on Duchenne's.
Mm. So, by the way, while we're on that subject, what are the side effects?
Well, the side effects of tamoxifen are about 80% of women have side effects, and basically it's what are called vasomotor side effects. So it's the principal side effects of menopause. So you have hot flashes, you have night sweats where you soak your bed at night. You have these rushes of warmth and that sort of thing. And those are about 80% of women on tamoxifen, and we have about 5% of women with endoxifen with those symptoms. There's some more serious side effects in like 1 in 5,000 where the uterus gets stimulated by tamoxifen. Endoxifen doesn't stimulate the uterus, it's one of the other molecules. We haven't done enough, we've only done about 750 patients with endoxifen. So we don't know in patients that we won't see the uterine effects, but we don't see them in any of the animal models. So it's cleaner from the very frequent but not serious side effects, and it's suggestive of the less frequent but more serious side effects, and we'll know as we go further into the clinical development.
So, what do you mean when you say that the uterus gets stimulated? Does it?
In with tamoxifen,
Uh,
about 1 in 5 or 1 in 10,000 women will have endometrial hyperplasia, and then about 1 in 1,000 of those will actually have endometrial cancer from the tamoxifen treatment.
mhm.
In postmenopausal women, it's more impactful than in premenopausal women because premenopausal women are constantly cycling the endometrium every month when they have menstrual periods. So
Yeah.
they don't see the stimulatory effect of tamoxifen. But in the postmenopausal women, you will see again about one in very rarely, but 1 in 15 or 20,000 will get an endometrial cancer from their breast cancer treatment tamoxifen. And we're hoping not to see that, but something as rare as 1 in 20,000 will take post-approval clinical observations to see it. What the new FDA commissioner calls real world evidence.
Hmm. So, tamoxifen has been been used for decades in breast cancer. What
Yeah.
problems are you trying to solve by giving the active metabolite directly?
Yeah. So a couple of them. Number one is because we significantly reduce the side effects of tamoxifen, the compliance or the ability of women to stay on the drug for 5 years after their surgery is projected to be much higher. One of the challenges is that once you're done with your surgery for breast cancer, you'll typically take tamoxifen or an aromatase inhibitor, another class of drugs, for 5 years to prevent two things: a recurrence in the same breast of that cancer that you had removed, and a new cancer in the other breast, because once you have breast cancer in one breast, you're at a higher risk for cancer in the other breast. So one of the purposes of endoxifen is to improve compliance during that 5 year period of time. But because we can go to higher doses, and because endoxifen has a second pathway called PKC beta, which is actually probably where it's working in Duchenne's, we can actually do a better job of killing breast cancer. So women whose cancers are getting bigger on tamoxifen, so they're effectively clinically resistant to tamoxifen, we can stop that tumor from getting bigger with endoxifen, and that's been shown in the clinic with small numbers of patients, but enough to show that that's a biologically relevant pathway. So I hope that answers your question.
Yeah, yeah.
Again,
So, see, you mentioned you mentioned metabolic variability. Can you explain why some patients respond differently to tamoxifen?
yeah, absolutely. Absolutely. So let's go back to how you get to endoxifen from tamoxifen. There's two different enzymes in the liver that do that hydroxylation, demethylation chemistry. Chemistries, A and B. And we get two copies of those enzymes from each of our parents. And there's two versions. There's a really good enzyme and there's a very poor enzyme. So women have sort of three phenotypes. Two very good enzymes from both parents. That's the best case. You're making a lot of endoxifen, from tamoxifen. One o each from your parents, not so good. And then you can actually have two poor ones. So the bottom line is that 40% or so of women taking tamoxifen don't make enough endoxifen to get any benefit. So they're taking this drug for 5 years. They're having side effects, and they are the women who will get a recurrence of their breast cancer because they're really not making enough endoxifen. And so that's kind of like a worst case scenario. Additionally, some drugs inhibit those enzymes. So if you're taking another drug at the same time you're taking tamoxifen, you can get inhibition of it. And that means you won't make enough endoxifen as well. There was a 6 year period of time. It was quite sad actually, where the 80% side effects from tamoxifen, somebody discovered, hey, if you give them Prozac, a serotonin reuptake inhibitor, the side effects go away, and they don't have the side effects, and it's really nice. So a lot of women went on Prozac who were taking tamoxifen because it prevented those side effects. And then about 5 or 6 years into it, we realized that what does Prozac was doing was inhibiting inhibiting the enzyme tamoxifen to endoxifen, so they weren't getting the benefit. So they were not getting the side effects, but they were getting recurrences of their breast cancer because they weren't making enough endoxifen. So that's one of the major clinical reasons why, if 40% of your women are not making enough endoxifen and 80% of your women are getting side effects, which again, 25% of those will stop taking the drug during the 5 year period because they just can't stand the side effects. It really, tamoxifen has significantly unmet medical needs, which is the code word for why a new drug should be taken, accepted by the medical community in a fast fashion. We've talked to a lot of doctors in the trenches with breast cancer, and they're chomping at the bit to get endoxifen.
Mhm. So your clinical studies include nearly 800 participants for with no maximum tolerated dose identified. What does that tell us about the safety profile so far?
Yeah, so again, this is kind of in the weeds, inside baseball conversation, but cancer drugs, because remember they started out with, as mustard gases, very cytotoxic chemicals. So the history is that you always determine what is called a maximum tolerated dose, which is a dose above which your side effects are so severe that you just don't want to go there, even if you could possibly save the patient, because you
Mhm.
probably, the side effects will, really take you down another path. And so all cancer drugs are driven in very early clinical trials, higher and higher doses to try to find a maximum tolerated dose. So you know what that bright line is, that red line that you can't cross. Um, in a very unusual, I'm not sure I know of another example of a cancer drug, endoxifen could not, a maximum tolerated dose could not be found, which again is very gratifying that even when you push the dose to get more and more efficacy, you don't see any side effect that is severe enough to say, I'm not going to go above this dose.
Mhm. So, Atossa is studying combination endocrine therapy, including the pairing of Z-endoxifen with CDK4/6 inhibitors. What is the combination therapy? Why is combination therapy becoming so important in oncology?
Yep. So maybe just a brief review of the pie chart of the kinds of breast cancer. They used to be just categorized in three categories, and for this conversation, I'm now going to make four out of them. So 75%, 80% are estrogen receptor positive. They're driven by estrogen. And then, so that's the vast majority of breast cancers. That's the vast majority that endoxifen works with. Then the two smaller categories that share the 25% are something called HER2, HER2 driven breast cancers. Are not driven by estrogen, driven by this other growth factor that's important. They're a little bit more aggressive than estrogen driven cancers, managed by antibodies now. Genentech and Roche were the first one with Herceptin, the first antibody drug for cancer that was so effective in the HER2 class. And then finally, there's a class called triple negative, which means they don't have ER, that's one. They don't have the progesterone receptor, that's two, and they don't have HER2. So that's a third category. Um, they're more prevalent in African American women. They're more prevalent in early age patients, and they're much more aggressive than either HER2 or ER breast cancers. ER breast cancers have a 90 to 95% 5-year survival. HER2 about 75 to 80, and triple negative can be down to 30 to 50%. So it's a pretty devastating category where you have to bring in chemotherapy, and it's got a lot of different drivers that make it a very challenging space. Now, in the ER positive space, there's actually two categories. There's sort of low proliferation, where you just need endoxifen to stop the estrogen driving, and then estrogen driven alone cancers that are high proliferation. So they have something else going on there. And that's a category where estrogen alone doesn't do very well. And about 10 years ago, this class of drugs called CDK4/6 inhibitors was brought to bear in combination with endoxifen to treat these high risk breast cancers. So they're identified at the time of diagnosis by something called Ki-67, which is a ratio of a fraction, a percentage. So it has a numerator and a denominator. So the numerator is the number of cells that are dividing, and the denominator is 100 cells or 200 cells or 300 cells. And so a low proliferation breast cancer that endoxifen is good for is something between 0 and 20% of the cells dividing. But high risk cancers where you have to bring in the CDK4/6 inhibitor are maybe 20, 30, 40, even up to 80% I've seen in some of the patients that I've been involved in the treatment. So in that special category of high risk but ER driven alone, the CDK4 inhibitors are a wonderful add-on combination therapy. So we've gone pretty aggressively into that when our generative AI. So again, back to this, where can AI help us? I went to one of the leading companies in generative AI for drug design called In Silico Medicine. Excuse me. And I basically charged them with saying, hey, look, we know out of the 20,000 blueprints that the cells make in general, we know what 5,000 are manipulated by endoxifen. Analyze all 4,000 drugs. What are the fraction of the 20,000 blueprints a cell makes that they manipulate, and which ones are synergistic with endoxifen? So these guys said, okay, that's a challenge. 4,000 drugs, 20,000 blueprints up and down. doing what's called a transcriptome analysis. They went away for 3 weeks. They came back and they said, hey, Lilly's CDK4/6 inhibitor called abemaciclib matches best with endoxifen. So 1 and 1 is not 2, but 4, 5, or 6.
Mhm.
We took that and working with Lilly very aggressively in a clinical trial program called iSpy run out of San Francisco by Laura Esserman. Within 100 days, we were giving it to patients who were at high risk of breast cancer. We've now done 80 patients in that space. We've reported on we've partial data at this point, not our complete data, but it's very gratifying. And what we saw in the in silico generative AI landscape, where 1 and 1 was not 2, but 4 or 5, in the patients, we're seeing again very gratifying results there. So that's, I think in the old days before AI, that would have been about a 4 or 5 year process to find that Lilly's drug was best. It took us 100 days. So that's an example both of the power of generative AI, and a particular example where we're partnering with Lilly's drug in a special class of ER positive cancers.
Mhm. So is the goal to improve effectiveness, reduce resistance, or both?
Everything, yeah, both. So the increased effectiveness in the primary cancer, where you want to kill them fast and early, is an important aspect. What we now know is that the aromatase inhibitors as a class, which block conversion of testosterone to estrogen, so postmenopausal women don't have any estrogen because their ovaries have shut down, but they still make testosterone in their liver and they still have an enzyme that converts testosterone to estrogen. So I remember in medical school, kind of raising my hand saying, why are you telling me that postmenopausal women that don't have any estrogen have an estrogen-driven breast cancer? Professor said, we don't know, but it just happens. And then we discovered this pathway where testosterone is being conversion after I finished med school. And so a class of drugs called aromatase inhibitors stop that conversion. So, but what we now know is that you give them every day for hopefully for 5 years. Within 1 year, about half of the cancers have said, I don't like, there's just a little bit of estrogen. I'm a cancer. So there are very characteristic mutations that occur in the estrogen receptor that make it resistant to estrogen. It's now always turned on. It's not just turned on by estrogen. And so that goes to the nucleus. It turns on so the process of treating breast cancer by aromatase inhibitors does what sometimes antibiotics do, which is it drives resistance. So you take an antibiotic and you have an antibiotic resistant infection. Now you're in a new space. And the same thing has happened in breast cancer. It's exactly the same genetics. Darwin is saying, yeah, this is how it works. You put pressure on a population, you put selection pressure on a population, and a new species or evolves out of that space. So again, one of the amazingly, I don't take credit for it, but one amazingly gratifying things about endoxifen is that it still continues to inhibit the mutated estrogen receptor. So it is not going to it's not selecting for estrogen receptor resistance because it works as well in the native estrogen receptor and in this mutated class of receptors. So that was data we reported last December, and the community was very excited about that because they're just now realizing that their own treatment, aromatase inhibitors for 5 years, are setting women up for recurrence. And about half of them end up with these estrogen resistant mutations in the receptor. And those are the women that have a recurrence. Again, we have a 90 to 95% success rate with estrogen-driven cancers, but we have a 5 to 10% failure rate. And we're now teasing out what that failure is. It's aromatase-induced mutations and then growth. So again, endoxifen is a wonderful drug because it's going to hit that space. It's going to hit both the wild type as well as this mutated state.
Mhm. So one thing that really caught my attention is the orphan drug designation for Duchenne muscular dystrophy. How did a cancer therapy lead to a potential treatment for Duchenne?
Back to the generative AI story. So look, I've done so much work with this company in silico medicine. I actually,
Yeah.
at my semi-advanced stage, I went back to school at Johns Hopkins to get a certificate in generative AI. And for a guy who, went to med school before computers, literally. I had to program Python for 3 or 4 months. So the power of generative AI comes to bear again in the story of how endoxifen gets repurposed for rare diseases. So going back to this 5-year interaction I've had with in silico medicine, I talked a little bit about what we did in terms of the transcriptome. So again, that's a fancy word for basically the 20,000 blueprints that every cell in your body makes every second, and then they control what proteins you make. So, basically you have a cell is a bag of a lot of things, but as much as anything, all of the machines in your cell that do things are proteins. And the proteins are made in a protein factory, and the protein factory uses blueprints, which are called messenger RNA, and they're RNA that comes from the nucleus. So the nucleus is the library of all 20,000 genes. And at any moment in time, they're making blueprints, and then the blueprints are being transcribed. So it's a very dynamic system, and it's massively beyond the capability of a single human brain to keep in one spot what's going up and down with 20,000 different blueprints, 20,000 different quantities of proteins doing activities at any point in time. Bring in generative AI, and basically it says, I can do this without breaking a sweat. So what we did was we said, well, what interesting things does endoxifen do for all 20,000 transcripts, not just the ones in the pathways that you can see clearly lead to cancer, clearly lead to breast cancer, and the like. And so about 2.5 years ago, we came back with this. I mean, they set up a very rapid meeting because they were so excited. They said, hey, look it. Endoxifen has this very unique activity. It's only been seen with a couple other chemicals, and it has very important aspects for a disease called Duchenne Muscular Dystrophy, and we think you should look at it, as soon as possible. And so we set up a meeting, we set up a Zoom meeting. I was in a Zoom meeting here. And so for the first time, they talked to us about the fact that there is a protein called utrophin, which is upregulated. That is, more blueprints are made, more protein is made when endoxifen is given to a skeletal muscle cell. Doesn't happen in all cells, only in muscle cells. I mean,
Hmm.
it's pretty interesting. So what does utrophin do? Well, utrophin is a protein that takes, so that you have the sarcolemma, which is basically like the dock or that sort of thing. And then there's a chain and an anchor that goes to your contractile proteins, the myosin proteins that cause your muscles to contract. And the anchor between this fixed sarcolemma or dock and the boat, or the proteins, during fetal development, so from a single egg until birth at 9 months, and for about 6 months afterwards, that anchor is a protein called utrophin. Same size as dystrophin, has some different properties. It's made on a different, it's not made on the X chromosome, it's made on another chromosome, not a sex-linked chromosome. So boys have it, girls have it. It's quite nice. And then in natural development, starting in the third trimester into the post-birth period, and then by 6 months to 1 year, all of the utrophin in this skeletal muscle is replaced with what's called a paralog of similar protein evolutionarily called dystrophin. And so in normal boys and wild type boys, they develop skeletal muscle, they grow, and this works just fine. In Duchenne's, there's, a couple thousand different mutations in this huge, huge gene, which prevent the anchor from being made in its full length. It's made in partial lengths. It can't do that linkage. You get inflammation, you get poor muscle function, and you get a gradual fibrosis where the muscles are replaced with fibrous tissue, so that by the time they're, getting on to be 10 or 12, they're transitioning into a wheelchair. And then in their 20s, they're having respiratory problems or cardiac failure, with a life expectancy that's, running around age 25 at this point in time. So what we hypothesize, I'm sorry, and then there's this one incredible exception, which, um, when I look at the creation of biology, I just am so in awe of how it all works. There's one set of muscles that don't replace utrophin with dystrophin, and that's your eyelids. So when you're blinking your eyes, you're using a utrophin driven protein. Every other protein in your body as an adult is using dystrophin. Why would that be? I have no idea, but it's just so fascinating how interesting this biology process is. So what we said was, okay, look at if we can upregulate utrophin, can we cause the muscle cells to drive it back into its fetal position? Because there's nothing there now. The dystrophin doesn't fill the gap, and we know that it's not working. We then looked and we said, hey, look at these boys are upregulating their utrophin about 20 or 30%. Not enough to be functionally all that useful, but maybe that's part of the delayed lifespan for these boys is the utrophin that they're making on their own. So we said, hey, let's build a model where we upregulate utrophin in a much higher extent, and could we reverse this disease? So I contacted some folks that had done clinical work that seemed suggestive that this would work in Switzerland and at Oxford University. They said, hey, you need to work with an animal model group in Switzerland that has a mouse model of Duchenne's muscular dystrophy. So instead of, a 10-year and a 20-year and a 25-year transition process, the mouse model compresses it into about 1 year. And let's see if endoxifen is effective in this model. And so we quietly, over a 2-year period of time, worked with this group, came up with, the study models. They have a model where they give the endoxifen to the pups to see if what happens to they become adults. They give it to the adults who are in a decline mode to see if it slows the decline. And in both cases, according to these investigators who've done pretty much all of the FDA-approved drugs for Duchenne's muscular dystrophy, endoxifen was best in class. That's their term, not my term, in restoring activity. And in fact, in the very simple model that they have of muscle activity in the mice, the Duchenne's mice with our drug run around the cage just like the wild type mice. But a more challenging test is they hang the mice sort of upside down on their cage, and the wild type mice will hang there for 5 minutes, 10 minutes. They'll never fall because their muscles are strong enough. But the Duchenne's mice actually begin to fall, in a couple minutes because they just don't have the strength. And in our work with the Duchenne's model, where we gave them, endoxifen from a young age, they held onto the cage forever, basically as much as the wild type mice. This data, which is very robust because, as I said, it's been used with every FDA-approved drug for Duchenne's, and according to these investigators, our drug is best in class. This data was taken to the FDA in the fall of last year to say, hey, we'd like to develop endoxifen for this pediatric disease, Duchenne's muscular dystrophy. What do you think of this data, and does this qualify for both what's called a rare disease designation and an orphan disease designation? And the FDA came back and said, this data is strong. We're qualified for both of those. So we've got you. They've given us the letter on that point. We're now in the process of designing a first in boy study, clinical study. We'll take it to the FDA. We'll get their permission to go forward. So our hope is by the second half of this year that we are giving endoxifen to boys with Duchenne's with the intent to change the quality of life for these boys, measuring typical strength measurements in our clinical trials, measuring measurements, around cardiac function, because again, one of the early clinical declines that is seen with boys with Duchenne's is heart muscle damage, cardiomyopathy, where they don't beat as much. That's one of the final last steps in the Duchenne's decline. Also the diaphragm. So if you can't breathe as well, you get pneumonia. I mean, that's coughing and being able to briskly breathe is one of the ways of preventing pneumonia. So both of those muscles we can look at with sophisticated MRI studies. Full disclosure, the gadolinium we'll use was I invented that. It was my first drug that's FDA approved of the 7 drugs I have that are FDA approved. But in any case, we're really excited about being able to contribute in this space because, as I said, I've now for the first time been interacting with the experts in Duchenne's, the scientific experts, the legal experts. The attorney that we hired last July actually was at the FDA as a young lawyer who wrote the rare disease law for Congress to pass. He was knighted by the Pope, or not
Oh.
knighted, but I'm sorry, given an award by the Pope for his contribution to the rare disease because Pope Francis, the late Pope Francis, was very much committed to the boys and the families of Duchenne's muscular dystrophy. He saw it as one of his strong missions during his time as Pope.
Mhm. So your research suggests that endoxifen may increase utrophin expression. For listeners unfamiliar with Duchenne, what is utrophin? Why is utrophin important?
It's kind of, repeating what I just said, which is that utropin is the anchor protein during fetal development and early life. Dystrophin is the anchor protein from about 6 months to a year for the rest of your life. And so if you don't have dystrophin because you have DMD, you're going to have this 20-year plus decline in muscle activity. If we can cause the boy's genetic system to reverse itself back to the fetal process where utropin is in place as the anchor, and it looks like the boys are doing that themselves in a small way, but not sufficiently to reverse the disease. Our hope is to be able to reverse the disease entirety and make this a chronic disease as opposed to a terminal disease.
Mhm. So for families affected by Duchenne, what does orphan designation actually mean? Does it guarantee the drug will be approved?
No, it absolutely doesn't guarantee FDA approval. The FDA standards for approval of regular regular drugs, orphan drugs, pediatric rare disease drugs is all the same. It's proving efficacy and safety and doing a risk-benefit analysis. That's always the same. What Congress realized, starting about 30 years ago, was that there were rare diseases under 200,000 incidents is the legal definition of a rare disease for adults or children. Uh, we're not getting the treatment, getting the attention of pharma because of the lack of financial incentives. So the orphan status from the FDA upon approval with standard criteria of safety and efficacy, the orphan status gives you 7 years of market exclusivity. So no matter what happens with patents and generic drugs and all that sort of thing, you know that you have a 7-year window to recoup your investment uh, because of the orphan status. The pediatric rare disease designation gives you that, and then it takes it a step further because the second, the rarer rare disease that Congress realized was the diseases in children. And again, there's reticence to do clinical trials in children because you have to consent via the parents. It's complicated, but nonetheless, these are real diseases that have a huge impact. And frankly, from my point of view, doing something for a 5 or 10-year-old compared to doing something in breast cancer for a woman, maybe 60s, 70s, 80s, those are different risk-benefit analyses because of just the natural longevity and potential contributions of I'm very excited to be in this pediatric space. So what Congress did was they came up with something called a rare disease voucher. So if you get a rare disease approved for a child, for a pediatric designation, which DMD is, and we have the designation from the FDA. If we get approval in that, the FDA gives us a piece of paper, which is called a voucher, which it's remarkable how pharmaceutical drug development works. All this piece of paper says is it gives whoever owns this piece of paper the right to a 6 month approval in the acceleration, a 6 month acceleration in the review process of a drug they have under development. So,
Mm.
but what that means for little companies like Atossa, and it's now been 20 years experience, we can sell that piece of paper for to a large pharmaceutical company who has this huge, multi-billion dollar drug because their CFO is saying, hey, every day, every day faster we can get this drug approved is multiple millions of dollars. So that voucher, and those vouchers have sold for between 100 and 400 million dollars to the large pharmaceutical companies. So that's a very strong incentive for companies to work in the rare disease space. It was realized by some economists, in the academia. Congress said, "Hey, this is a good idea," and it really shows the sort of the power of capitalism in the markets to reorient companies into rare disease spaces in children that I think otherwise would not be addressed. And so we have many pediatric rare disease approvals for which this process is the beneficiary.
Mhm. So, drug development can take many years. What are the biggest hurdles between promising science and approved treatment?
Well, my wife, who's actually a cancer biologist, says I'm crazy to be in this field because there's 9,999 ways a drug can fail and only one way it can succeed. So it's a huge process of attrition where you have promising molecules or promising biologics in animal models, and then they fall off the wagon between discovery and model testing and first human testing, and then phase one. There's a huge attrition where you're just looking at safety in phase one, normal volunteers, and then phase two, your first introduction to the disease state, where hopefully you're seeing efficacy, hopefully you're seeing a lack of safety issues, and then phase three, which is typically very large trials. They're usually some fraction of the total population. So, if you're developing a drug for a million people or 500,000 people, you're going to have to do a trial maybe of 5 or 10,000 people. So in any case, those all of those are hurdles for drug development, and biology is not chemistry, and chemistry is not physics, and physics is not math. That's my hierarchy of knowledge and understanding in the world. So maybe with generative AI, actually, I mean, the reason I'm so excited about the next 5 years is maybe generative AI can take this black box we call biology and open the hood and look inside and say, hey, there are some rules, there are some equations we can design that describe what's going on here. Maybe we can elevate biology towards chemistry, towards physics, towards math, which is where I love Elon Musk. I think he's a genius,
Mm.
but he is an engineer and he is working mostly with math and physics and not with chemistry and biology. So if we could get my area of biology into his space, maybe we could do some of the cool things he's doing.
Mhm. It's interesting where these drugs go. It's like they could how they become a drug for something completely opposite field. Doctors don't even talk to each other. It's how does this happen? It's
Well, I mean, it happens again because biology is such a black box.
yeah.
That's proof, every time that happens, off-label use. Um, now, I would say that doctors are pretty good about reading the literature and reading an article and saying, hey, this is an interesting study. Maybe it's a small study or something of patients with a disease that I wouldn't have thought this drug would be useful for, but it's off-label use. Maybe the pharmaceutical companies, sponsored the trial. Maybe the doctor on his own said, "Hey, I'm enough of a scientist. I'm seeing this pattern with a couple patients. Let me do 50 or 100 patients to get some statistics around this." And doctors will adapt that. Now, in oncology, oncologists are the biggest and best experimentalists of all of the classes of physicians. So, I haven't done the analysis. I probably should, so I can talk fluidly about it, but a very high percentage, and I'm going to say at least a third and maybe a half of drug use in oncology is not FDA approved uses, but off-label use. There's something called the NCCN network, the
Hmm.
National Cancer Care Network is what the NCCN stands for. They meet every 6 months and they put out guidelines, which is basically a recipe book for how to treat patients because they are authorities on off-label use. And they come up with these algorithms of, well, if the patient has this and the patient is this age, all these properties, then this is the path we recommend use. And again, a large percentage of them, a third to a half, I should get an official number so I can use it cogently, but a large percentage are off-label. And so uh, other disciplines don't do that as rigorously as cancer oncology. But I'm going to repeat myself now. I think doctors are pretty good at finding these off-label uses. And I think, again, I'm really encouraged by what the new FDA commissioner is doing, Martin McCary. Because he came from Johns Hopkins. He came from being a surgeon. He came from doing clinical trials where IRBs were slowing him down and all these sorts of things. So he is quite clever and in his leadership and what he's doing to modernize the FDA and make use of some of these AI tools and to make use of rapid mechanisms of getting things approved faster. He just did a speech, a wonderful speech where he compared U.S. versus Chinese processes for simply getting, through what's called the IRB, which is permission to do clinical trials. U.S. it's 350 days, and China it's 20, and their now goal is 60 days, just to begin a trial. So we need to modernize. We need to be quicker. We need to take advantage of the modern tools we have, and it's gratifying to see that happening, inside HHS and the leadership there under Secretary Kennedy.
Hmm. So, rare disease communities often worry about access to clinical trials. What advice would you give to families who want to participate in research?
So I'm a neophyte in this space, so I don't want to be giving too much advice. But I can tell you after going to the muscular dystrophy meeting down in Orlando last week, yeah, seemed like a long time ago. Time flies,
Eh.
and being at a forum up in Boston, that I think families can find resources and communities. I mean, I was just impressed with the community advocacy and coordination, via the internet, via these groups that you can find on the internet, for finding clinical trials, for getting support, for all of those sorts of things. So DMD, has a lot of support on the internet. And endoxifen also works in a disease called McCune-Albright syndrome, which is precocious puberty in young girls. It blocks the
Mhm.
estrogen. I mean, these poor girls have a lot of different things going on. But one of the ones we can address is that they begin to have periods, when they're 4, 5, or 6 or 7. Kind of a devastating psychological thing for these young girls. We believe we can stop that with endoxifen. We're going to go into the clinic with that hopefully soon. The other thing is that the high estrogen they have, the artificially high estrogen because of their condition, they close their bones early, so they're very short stature. And so again, we believe endoxifen can help keep the epiphyses open to allow kind of normal stature recovery. So but when I looked at the mechanism in the biology, of this, and then went myself to the internet, there's a wonderful organization for Duchenne's muscular, for misspoke, for McCune-Albright syndrome, and we're in contact with the organization there and working with them to try to identify patients and physicians here. So I think the communities are there. I mean, that's the best I would say to the patients. I'm finding them sort of in real time now myself and finding it, pretty easily. So that's what I would say to any new patients coming along that might be listening to your podcast and,
Mhm.
just beginning their journey with a child with one of these conditions.
Mhm. So, how do companies decide which rare disease to pursue?
Oh, it has to be driven by biology. It's,
Mm.
90% driven by biology. You can't. The rare disease space is challenging for lots of reasons. And so you have to have really good biology to begin with.
Mhm. So you also worked with at the intersection of biosecurity and national preparedness. How does drug innovation intersect with national health security?
Yeah, so um, I'm a scientific nerd, I guess is the basic place to start there. I did some work in 2020 on an interesting pattern of where all the patients who were getting COVID early in Wuhan, China were located. They all were located along one of nine subway lines. All the hospitals were on one line of subways in Wuhan in the first 6 weeks. And that line connected the Wuhan Institute of Virology that's doing research on coronaviruses and the airport where Wuhan had, 15 different nonstop flights around the world. And then the outbreaks around the world were coinciding with all these cities that had the nonstop flights from Wuhan. So I published this paper, I put it up on the internet, I forget about it. And then I get a call from Secretary Pompeo's office, in the State Department in the United States saying, "Hey, we'd like to talk to you. Um, are you a spy? Are you a Chinese spy? Why do you know this? Why did you do this?" Because, our own scientists in the intelligence community at Lawrence Livermore, um, who have confidential information, came to the same conclusion after you did. So I started helping them in that space, and I've testified in Congress a couple times, and then was appointed a senior fellow at the Hudson Institute on the intersection of AI and biotechnology and national security. So um, I think the 70/30 of what I do there is the fact that SARS-CoV-2 was designed in a laboratory by humans. It was about 1% lethal. And the 5-year development in generative AI has now allowed us to use that tool to make viruses that are much more lethal, much more transmissible, have many dangerous properties. And so I've advised some folks in Congress and that in the intelligence community to to address this. We say, it's a national security issue that countries around the world can use their generative AI to challenge us with new infectious agents that we don't know about. The second factor, though, that is also very important is the national security aspects of our pharmaceutical supply chain. So, for perhaps lots of good economic reasons in the 90s and the 2000s, where we brought China into the World Trade Organization and expanded globally. Everyone wanted to have a global footprint. Um, having 90% of your essential pharmaceuticals, so there are about 20 pharmaceuticals that are called essential by the WHO, and every country has to have them in their supplies for emergencies and distribution in their warehouses and that sort of thing. Like 90% of the essential pharmaceuticals are made in India or China, and not made in the US. So one of the things that I'm talking about that I think is important is the concept is how do we repatriate the manufacturing of these pharmaceuticals in a way that makes our supply chain sovereign to the U.S. So in a worst case scenario, someone doesn't cut off our supplies. The analogy I keep telling people is, look at if we'd had to go into World War II with having steel not made in Pittsburgh but made in Germany, we would have been in trouble because our ability to enter World War II was based on our ability to make steel in Pittsburgh. And
Mm.
so, not having a pharmaceuticals is a national security concern. Going back, one of the exciting things you're hearing a theme running down this podcast is that the use of generative AI and robots in the pharmaceutical manufacturing could allow, in a hypothetical future U.S. manufacturing plant, the ability to make it extremely cost effective for making pharmaceuticals in the U.S. So it could not only be the right thing to do from a national security point of view, but it could actually be economically viable, which is again one of the things that drives the private sector a lot more than maybe patriotism or other aspects. But being able to make money with a shorter supply chain that can get broken, like it did during COVID, repatriating these things in U.S. based factories with a generative AI robot continuous manufacturing cycle is a pretty exciting idea.
Mhm. So looking ahead, what emerging technologies and drug developments are you most excited about?
So look, I mean, let me again, I want to kind of go from, sort of a national view on this. So,
Mm,
and I've gotten a little bit involved here. So I think um, what Secretary Kennedy is doing at HHS, and especially Dr. Jay Bhattacharya at NIH, who we both were Stanford professors in different eras, but nonetheless, what Jay's doing at NIH is really going to be revolutionary because he has seen the power of the generative AI possibilities in biology, and because he has the largest footprint in biomedical research as director of NIH, he has ability to deploy resources at a level that no one else, Gates Foundation, the Wellcome Trust, China government, no one else has the footprint that he has in biomedical research. And he's very aware of the power of generative AI, and he's very astute in deploying these resources in ways that are going to be synergistically valuable for US. The window of opportunity to take the lead in generative AI and biomedical research is now and over the next, I want to say 5 years, but it may be as short as 2 years. So we are in a foot race or or more than a foot race with other countries to take the lead in this. And first mover advantage is going to be critical. And so I'm a strong component in making sure that the U.S. wins that foot race. And so, anything I can do to help in this context. But I can tell you reassuringly that our current administration, especially under what's going on at NIH and the various institutes there, has the best interests of this, and I think has a plan that will be successful and make us sort of win this important foot race.
mhm. So after decades in medicine and biotechnology, what keeps you motivated to continue to do this work?
So I mean, full disclosure now. I invented gadolinium for MRI. It's been used in about 80 million people. I sold my company at age 39. And I was moving, living around Europe as, just sort of enjoying life, kind of retired at 39, enjoying life. About a year into it, I realized I'm not a hedonist. I mean, I really like my work. I really enjoy my work. I feel like I can make a contribution. So I went back to work. And, despite the fact now it's, been a number of decades since that time, I love being able to contribute. I feel it's a privilege to be able to do that. We're only given so much time. I have a lot of gratitude for every day I'm given where I can make a contribution. And I have young kids, and including a young freshman in college who I want to set an example for you can do.
Okay.
So being at the intersection of helping people, the national security aspects that our government has is very gratifying. And as my wife says, I enjoy my work.
Hmm. Yeah, it shows. Let's see, many of our listeners are patients or caregivers living with rare conditions. What um, message would you like to share with families who are waiting for new treatments or to be developed?
Well, I think it's a couple things. So if it's a if it's a rare condition that's named that has how do I say it, maybe 500 to more people in the country, the thing you should do is try to plug into the companies or academics or other entities that are already in that area developing drugs and and looking for ways to innovate. I have to say that if you have a rare disease, which is which maybe doesn't have an advocacy group and a website and all those sorts of things, I have to tell you, I would immediately contact both the FDA Office of Rare Disease and the NIH Office of Rare Disease and the NIH Hospital. Because again, what NIH is doing is they're working across silos. So one of the dangers, inside the government is everybody has a silo and they don't talk to each other, but not in this administration. So NIH and the FDA are collaborating very carefully, and we have a new guidelines around ultra rare disease, which is exactly what I'm talking about. So there is now an established pathway for the FDA to deal with diseases where maybe there's one patient or there's two patients or there's three patients. So this is an incredibly rare time. I'm using the word rare a couple different
Yeah.
ways, but for those kind of patients to be coming to the fore. So if you have a disease where you don't think anyone else in the world has it, this is the best time. I'd contact the ultra rare disease group at the FDA. I'd contact NIH. NIH does more clinical trials in cancer or in rare diseases than any other group combined. The clinical trials are typically done, with, the care is provided, the cost of the care is provided inside the hospitals within the NIH or organization. So that's my advice.
Hmm. Yes. Is- uh, there's one message that's echoed through this: you are not alone. Um, even the ultra rare conditions, there's at least two of them I know of that are getting first treatments, but they're having to spend millions of dollars for it. So that's the downfall of it, is they have six people, and so they have to spend more money. Yeah, so it's in order to get these drugs developed, unless they go overseas, and so that's one way out of it. Um, a lot of the other countries will do this for less.
So,
Um,
certainly clinical trials are extremely expensive in the US. There's no
yeah.
question. And that's a systemic problem, and it's from rare disease to common diseases. It's become quite an industry. For a while, it was an advantage for a small company because there are people that could help you all over the place. But the clinical trial industrial complex has now become, such an important silo inside of Wall Street, effectively,
Hmm.
that it's hard to do clinical trials without a lot of money.
So why is the cost so expensive?
It's become a business onto its own.
Hmm.
Um, Number one, it's going to become a business onto its own. Number two, before the current FDA commissioners, and I don't want to, but there's been an arc of the FDA and probably pharmaceuticals in conjunction making clinical trials more and more complex from a statistical point of view, from a record keeping point of view. Um, the conversion from paper clinical trial records to computer sounds good, sounds wonderful, all sorts of things. There are whole companies whose whole business is doing clinical trial records in a computer. Um, I love it, but on the other hand, just a 20-page report. So there's sort of money being sprinkled all along the process. The FDA now only accepts submissions through a particular website, and they have to be formatted in a particular way. So there are particular companies that will, because it's expensive to format your submissions in a particular way to fit the FDA's particular website. So, um, I mean, the sort of business aspect of clinical development probably is one of the contributors. The complexity of the clinical trials and the expectations, and I think we do have as a people um, join in the process. Uh, Clinical trial development is a continuum between clinical trials, FDA approval, and then real world experience. And I think, frankly, the tolerance of the community for drugs that have side effects that are rare, that can't be seen in clinical trials, but get seen during the real world experience. I think people's tolerance for those kind of side effects is lower. So the FDA and the pharmaceutical companies' response is, well, let's make our clinical trials bigger because statistically, the only way you're going to find rare things is to do more people in the primary population of trials. So I think everybody has a hand in the pie of why this is also expensive.
Hmm. So, Dr. Quay, thank you so much for joining me today, and helping us better understand the science behind drug development and how innovation in oncology may also help doors for rare disease communities like Duchenne Muscular Dystrophy. It's always encouraging to hear about research that has potential to improve lives across multiple conditions. To everyone listening, if you enjoyed today's episode, please subscribe to this podcast, Rare Connection, and follow my YouTube channel, Rare Chef, where where many of these conversations are also available in live format, in video format, live also, but not as many. If you if you're watching on YouTube, I'd love to hear your thoughts. So please leave a comment below. And if you're listening to the audio version, you can use the send me a text link in the show notes to share your feedback questions. You can also follow Rare Connection on Facebook, X, and LinkedIn, and stay up to date on upcoming episodes featuring patients, caregivers, researchers, and advocates from across the country, across the rare disease community. And as always, if you or someone struggling and needs support, help is available. In the U.S., you can call or text 988 to reach the Suicide and Crisis Lifeline. If you are outside the U.S., please check your local your local crisis resources. Thank you again for listening, and I'll see you next time on Rare Connection.